Hi, my name is Akin Hyatt.
Hi, my name is Lauren McCaffrey.
And this is our Lab 11 video summary. We rely on vision more than almost any other sense, but how does the human eye work? After passing through the cornea, light enters the pupil and the lens. The amount of refraction can be controlled by contracting the ciliary muscle. The light then focuses on the retina where photoreceptors transmit an image to the brain. Like in other labs, the eye is an optical system which we can characterize using specific figures of merit. Similar to having a dominant hand, we also have dominant eyes with which we prefer when focusing on objects. To test which eye this is, we limit the field of view of the eyes to a small target. First, we focus on some object in the target with both eyes, then close one eye at a time without moving. The image formed by the dominant eye will be identical to the image formed by both eyes and will not see a shift. Note that this procedure works for any kind of sighting mechanism and is not limited to a circular cutout. Shown here are the results for our eyes. As you can see, both our right eyes are dominant, which we expect given that we are both right-handed. Since the optic nerve must connect to the eye somewhere, a region on the retina exists where there are no photoreceptors, called the blind spot. To find it, we drew two dots on a piece of paper a known distance apart. Focusing on one of the dots with one eye, the paper is brought closer until the second dot vanishes. The angular position of the dot at this position represents the angular location of the blind spot relative to the center of the FOV. We measured this angle to be about 15 degrees.
In addition to the blind spot, we can examine our eye's entire FOV. The iris defines the maximum angle subtended by the eye, and unsurprisingly, our FOV increases when we consider our ability to rotate our eyes and head. To measure the monocular or single eye, biocular, and overall biocular, including eye movement FOV, we used 2 meter sticks and rotated them until they were at the limits of our FOV. Then we measured the angle between the meter sticks with a protractor to get our full field of view for each case. Our results somewhat match the typical FOV ranges, and differences are likely a result of the difficulty using meter sticks in this approach.
One of the most important characteristics of vision is the quality of images produced. In this lab, we focused on the eye's ability to separate fine lines similar to MTF and LSF metrics for other optical systems. Acuity is typically measured using a Snellen chart, in which a subject stands a known distance away and attempts to read smaller and smaller rows of letters. Given the standard angular dimensions of each letter, acuity is quantified relative to what an ideal eye can resolve from 20 feet away. With corrective lenses, our results for each eye are listed in this table. Note that the quality of one's eyes depend heavily on the amount of aberrations present in their eyes, as well as natural defocus and medical conditions. Another test to determine visual acuity is the FACT test, which qualifies one's ability to resolve successively faster spatial frequencies at varying levels of contrast. The test is designed such that healthy eyes lie in the shaded portion of the figure to the right.
We also analyzed the color vision of the eye. 2 photoreceptors exist in the eye, including cones and rods. Rods used in scotopic situations have high density near the fovea, and the cones are dispersed along more of the retina. The peak wavelength sensitivity of the cones and rods varies to accommodate different lighting conditions and uses. Photopic vision peak sensitivity is red-shifted, which makes sense provided that blue, green, and red cones exist. We tested our wavelength sensitivity limits by inputting a broadband source into a grading monochromator, which isolates frequencies as shown. We scanned over the wavelengths present in the source until we could no longer see light. Our results were close to typical wavelength limits of 380 to 700 nanometers, and our measurements were likely impacted by our experimental setup, the power spectrum of the source, and tolerances on the monochromator reading. Similar to other imaging systems, the eye also has temporal limits. The flicker fusion frequency, or FFF, is the maximum frequency which is detectable by the eye and does not appear as a continuous signal. This concept is similar to aliasing or undersampling of a signal and is taken advantage of in film and television. To test the wavelength and power dependence of FFF, we drove a resistor LED circuit with a sine wave at varying frequencies and amplitudes, then identified the maximum frequency which we could still see blinking by scanning over frequencies from 25 to 70 Hz, which included typical FFF values. We did this for a red, green, blue, and pink LED. Our results demonstrated that an increased current applied to the LED and therefore power emitted allowed us to perceive flickering at higher frequencies, and we could detect green to the highest frequencies given that our peak sensitivity is near 550 nanometers.